Hello everyone, we are back with a new chapters, and as you can see, today's chapter is carbohydrate, which is in fact the most important chapters and the most interesting chapters as well. So without any further delay, let's begin. As we all know that carbohydrate is almost present in almost all the foods that we consume. It actually provides you flavor, color, and texture to the food. It actually is also called as carbohydrates can also be called as saccharides or say sugar units, right? And it is also called as like polyhydroxyaldehydes or ketones. It can also be defined as polyhydroxyaldehydes or ketones, or you can say it is a product that it produces on hydrolysis. Suppose when carbohydrate undergo hydrolysis, it will give a product like polyhydroxyaldehydes or ketones. Simple as that. And the energy that is provided by carbohydrate is 4 calorie per gram. Okay, very simple. 4 calorie per gram is energy provided by carbohydrates on digestion. Okay, now carbohydrates can actually be divided into or can be say classified into different types like monosaccharides, oligosaccharides, and polysaccharides. Okay, carbohydrates can actually be divided into monosaccharides, polysaccharides, and oligosaccharides. So starting with the first, that is monosaccharide. This monosaccharide, it is actually the most basic one. Why? Why monosaccharide is actually the most basic one is because it is having only one sugar unit or say one saccharide, because of which it is most basic. Okay, and examples are like polyhydroxyaldehydes, which is called as aldoses, and ketones, which is called as ketoses. Okay, so these are basically the examples. Now I have given here some examples of hexose sugar. Okay, I have given here some examples of hexose sugars. Like you can see the glucose, the galactose, and the mannose, and the fructose. These are actually the most say these are the examples of hexose sugar. Okay, so if you see here the fructose I have mentioned here, actually let me tell you one thing about the fructose is it is the most common ketose sugar in plants. What is that? This D-fructose is the most common ketose sugar in plants. Simple. Now, if we see here, I have given starch to glucose. What is this? See, when starch undergo hydrolysis, or say when starch degrade, it will give glucose. Okay? When starch degrade, the product is glucose. And when this glucose, and no, no, see, and when this glucose isomerase, this is an enzyme, okay, it acts upon glucose, the product will be fructose. Simple as that. Means when this glucose isomerase, that is an enzyme, act on glucose, the product will be fructose. Simple as that. Okay, and this glucose and fructose actually is used as syrup. Okay, glucose and fructose can actually be used as syrup. Now there are some examples of rare sugar, like you can say the allose, the piscose, the decatose, and the talose. So these are the examples of rare sugar, or say rare monosaccharides. Means they are not that much available. Okay, it is not that much found in most of the foods. Okay. Okay, so going to next slide, as we have now is disaccharide. Disaccharide, as we can see or say by its name, that it is two, okay, two sugar units. Simple as that. uh See, before explaining this one, let me tell you that as I have told you, there are three classifications can be done to carbohydrates. That is monosaccharides, oligosaccharides, and polysaccharides. Okay, under oligosaccharides, these disaccharides come under. Okay, actually, you have to understand before this is that what is oligosaccharides. First of all, let me tell you that oligosaccharides is it is to have 2 to 20 monosaccharide units. How much does oligosaccharides have monosaccharide units? 2 to 20. Simple as that. Okay, and oligosaccharides have glycosidic bond. What bonds do it have? It have glycosidic bond. And these oligosaccharides can actually be divided into different types like disaccharide, trisaccharide, tetrasaccharide, and pentasaccharide. Okay, disaccharide can actually be divided or say classified into disaccharide. I mean, oligosaccharides can actually be divided into disaccharide, trisaccharide, tetrasaccharide, and pentasaccharides. Okay, so first thing first, disaccharides. Okay, now this disaccharide actually can be divided into two, or say broadly classified into two. One is reducing and the other is non-reducing. But one is reducing and the other is non-reducing. Now this, whether it is going to be reducing and non-reducing, actually depends on the anomeric carbon in glycosidic bond. Okay, what does it depends on? It depends on anomeric carbon in glycosidic bond. Simple. Yes. Now, if I talk, or say, if I tell you about the examples like reducing, what is the examples of reducing disaccharide? Actually, is maltose and lactose. Okay, reducing disaccharide is maltose and lactose, and non-reducing is actually the sucrose. Okay, non-reducing is actually the sucrose. Now, as you can see, I have given here some of the examples like on hydrolysis. When this disaccharide undergo hydrolysis, it can actually result in the same, you can see here, it can result in the same uh, monosaccharide, or it can also result in different monosaccharides in it. Okay, like if maltose undergo hydrolysis, it will give glucose and glucose. Okay, and if sucrose undergo hydrolysis, it will give you glucose and fructose, and lactose undergo hydrolysis, it will give you glucose and galactose. Simple. Now, talking about maltose, or say maltose, right? It is actually formed by starch hydrolysis. Okay, how is maltose forms? It is the product of starch hydrolysis. Okay, and this maltose is actually produced by barley malting. Okay, as I mentioned here, it is produced by barley malting. And actually, let me this barley malting. This is actually commercially used to produce maltose. Commercially, productions of maltose is done by barley malting. Simple as that. Now this maltose actually used in beer production also. Okay, simple. It's just a matter of you have to remember it. Okay, and talking about milk, this I mean lactose. This lactose concentration in milk is almost 4 to 9%. Lactose concentration in milk is around 4 to 9%. And this lactose is actually the primary carbohydrate source. What? Lactose is actually the primary carbohydrate source, because for it is actually primary carbohydrate source for child. Okay, because they feed on milk, so this is the only source for them as for carbohydrate. Okay, and if we talk about some of, see, this one is, I have written here, this the galactose and the glucose. What is this? Actually, these are the product of hydrolysis. When lactose undergo hydrolysis by an enzyme, there is lactase. Okay, or say beta galactosidae. Okay, lactase, when this lactose undergo hydrolysis in the presence of an enzyme called lactase, it will give you a product called the galactose and the glucose. Simple as that. Okay, coming to C, there is sucrose. See this sucrose, this will give you a this will give you a product that is alpha D glucose and beta D fructose on hydrolysis. Okay, alpha D glucose and beta D fructose is the product. Now this sucrose actually is linked by reducing ends. Okay, what does reducing end actually means over here? Please listen to me carefully. It actually means reducing ends means it is having free aldehyde or ketone group. Okay, whenever we say this is a reducing ends, it basically means it is having reducing end and or aldehyde end, free aldehyde ends a group or ketone group. Okay, simple as that. Now this sucrose, where it is formed, sir, it is formed in cytoplasm. Okay, sucrose is actually formed in cytoplasm, and this sucrose is actually a quick source of energy. Okay, sucrose is actually what? It is the quick source of energy. Okay, now as I have told you before, that oligosaccharides can actually be divided into four types, or say classified into four types. They are disaccharide, trisaccharide, tetrasaccharides, and pentasaccharides. This actually is not disaccharide. Okay, this is actually a part of oligosaccharides. Please forgive me as I have written here mistakenly. Okay, this is a part of oligosaccharides where trisaccharides, tetrasaccharides, and pentasaccharides are all there. If you see trisaccharides, is examples, trisaccharides example is raffinose, that is fructose plus galactose plus glucose, and tetrasaccharide is stachyose, that is 2 galactose plus glucose plus fructose. Okay, these are the products that is used in undergo when it undergo hydrolysis, it is the product. Okay, now tetra means 4, right? So 2 plus 1 plus 1, so it is actually 4. Okay, that means tetrasaccharide. Okay, coming to pentasaccharides, example is barbacose. There is 3 galactose and glucose and fructose. Okay, so 3 4 5 penta, right? 5. Okay, it's like this. Okay, now talking about polysaccharides. Poly means it is having many. Obviously it is having many monosaccharide units. What? Poly means many, right? So it is having many monosaccharide units. If we see here, poly means it is having more than 20 monosaccharide units. Poly means it is having more than 20 monosaccharide units. Yeah, so this polysaccharide actually can be divided into, or say classified into two types. Okay, if you see here, one is starch and non-starch. Okay, first of all, let's try to clarify this one. What is starch and what is non-starch? See, starch actually will give energy. Okay, starch is energy source. What about non-starch? Non-starch is anything that is non-digestible. Okay, non-starch is non-digestible, and it actually maintains intestinal health. Since non-starch is non-digestible, it will actually maintain intestinal health. Okay, and the other I have mentioned here is homoglycans and heteroglycans. What actually is this? Homo, we all know it is same. Hetero means difference. Okay, so what actually is this? Homoglycans means same sugar, heteroglycans means obviously different sugar, right? See, talking about polysaccharides, actually there are two basic chain conformations. There are two basic chain. Okay, one is ribbon-like and the other is hollow helixes. Yeah, you can see I have mentioned here ribbon-like example is beta 1 for diglucosyl linkage that we have, and hollow helixes means they will have alpha 1 for diglucosyl linkages, right? And linkage, talking about linkage, it is having beta 1 3 diglucosyl linkage. Okay, what does linkage does it have? It is having beta 1 3 diglucosyl linkage. Simple as that. Okay, now as you can see over here, um please, yeah, this is actually this beta 1 3 diglucoside linkage is actually the hollow helixes. Okay, examples of hollow helixes, please note it down. Okay, hollow helixes examples actually is beta 1 3 diglucoside linkage. Now talking about glucans actually is a polymer of glucose, and fructose, yeah, fructose, it is a fructose. Okay, yeah, it is a fructose, not fructans, it is fructose, and fructose is actually the polymer of fructose. Simple. Now dextrins. What is dextrins? Very important questions, right? What is dextrins? uh See, it is actually the product of starch. Okay, when starch undergo breakdown, or say when starch breakdown, the product that you will get is actually dextrin. Simple. And let me tell you that how does it undergo or say breakdown? I mean, how starch breakdown takes place? It is because of um, say your enzyme amylase, or because of the dilute acids. How starch breakdown? It is because of amylase or dilute acids. Simple as that. Now talking about this intermediate, see, as I have written here, intermediates. These are intermediates that formed. Okay, like you can say soluble starch is blue color, amylopectin is violet color, and artrodextrin is red color, and astrodextrin or say acrodextrin is no color. In fact, this color, how do we got? How can I say this is that color? This is that color, and how actually we got this color is actually by iodine coloration. Okay, this iodine coloration will give you this color. Simple as that, nothing much to explain here. You just have to memorize or say remember. So yeah, pectin. See, pectin is one polysaccharide. Okay, pectin kya hai? What is pectin? It is actually polysaccharides, and it is actually composed of galactouronins and rhamnogalactouronins. Okay, this pectin is composed of what? Galactouronins and rhamnogalactouronins. Okay, rhamnogalactouronins. Okay, and what is a pectin backbone? Is beta 1 for the galactouronic acid. What is a backbone of pectin? Is beta 1 for the galactouronic acid. Okay, and pectin is actually water soluble. What is this? Pectin is actually water soluble. Let me tell you one more thing is that This pectin actually can be divided or say classified into two more types. Pectin can be divided into two more types or say classified into two more types. One is high methoxy pectin and the other is low methoxy pectin. Okay, so what is high methoxy pectin? High methoxy pectin means you can say that it is having more than 50% methylation. What does it have? It is having more than 50% methylation. And this high methoxypectin, you know what? It is water soluble. Okay, high methoxypectin is water soluble, and you know this high methoxypectin form gel in acidic condition. High methoxypectin it forms gels under high acidic condition. Talking about low methoxypectin form gel in the presence of calcium, means not in acidic condition, but in basic condition or say calcium under calcium presence. Low methoxypectin form gel. Simple as that. If you're not understanding what I'm explaining, please go back, rewind back the videos and watch it again. I want you all to understand what I'm explaining. Okay, I am spending my lot of time, lot of energy to make you understand as much as possible. So please rewind it back and try to understand try to understand what I'm explaining. And if you have any kind of confusions and doubts, please let me know in the comments below. Okay, so low methoxypectin actually it does not need sugar to form gel. Okay, low methoxypectin does not require gel to form. I mean, it does not require sugar to form gel. Now, these talking about this pectin, it is also used as gelling agents. This pectin is also used as what? Gelling agents, right? Talking about starch, this is very important. That I want you all to understand. Okay, please listen here to me carefully. I will try my best to be simple as to be, I will try my best to be as simple as possible. Now, starch is also called as what? Amylum. Starch is also called as amylum. Okay. Now, this starch actually have large number of glucose units. What? This starch actually have large number of glucose units, and they are joined by glycosidic bond. Starch have large number of glucose units and they are joined by glycosidic bonds. Simple. And this starch actually is produced by green plants. Okay, this starch is actually produced by green plants, because you will find starch in plants, not in animals, right? What is found in animal? It is glycogen. Okay, that we will discuss later. Now, uh starch is one form of storage carbohydrate. Let me tell you this one, everyone knows that one obviously. Now this starch, it is composed of two polysaccharides. Starch is composed of two polysaccharides. So what are those two polysaccharides? It is amylose and amylopectin. Okay, starch is composed of two polysaccharides and they are amylose and amylopectin. Let me tell you that this amylose is 15 to 20% and amylopectin is 80 to 85%. Simple as that. Okay, amylopectin is 80 to 85%. Simple as that. Now this starch isolation is done by centrifugation. Okay, how is starch isolation done? It is done by centrifugation. So what is centrifugation? Anything where centrifugal force are being used is called centrifugation. What is centrifugation? Centrifugational force. Everyone knows it. I hope so. Right? There's nothing much to explain over here. Uh yeah, so yeah. What happens is that if there is more cross linkage in your starch, it will be more tolerant to acidic condition, because it is having more cross linkage. That means it is more strong. Okay, and because of which it will be more tolerant. It will be able to tolerate high acidic conditions, and there will actually be less breakdown of starch. Okay, since there is more cross linkage, there will actually be less breakdown of starch. Simple as that. I think that's all with starch. Let's go to the next slide. Okay, so yeah, so this is the end of our today's topic. I hope all of you understand or say I have understood what I've explained. Please let me know if you have any sort of confusions or doubts that needs to be clarified. And if any one of you, or say if all of you want to participate in the mock test series, please let me know. I will actually give my WhatsApp details, WhatsApp numbers, and my Telegram group links, and yeah, all sort of things you'll get in the description below. And yeah, we'll meet in next video. And that's all for today. Have a nice day, and bye bye.